Hey there, science fans. This is Dr. Cummings from Point Loma Nazarene University. This video is the first in a series on how to read the primary literature in science, and in particular in microbiology, which is my field. In this first video, I really just want to kind of introduce you to the idea of what a primary research article is, and then the anatomy of the paper itself so that you can find your way around it. In subsequent videos, we're going to talk in more detail about what you can get out of each of the different components of a journal article, and maybe how to tackle some of the trickier parts. So first of all, I just want to define what we mean when we talk about a journal article, or we say the primary literature, the primary research. These professional journal articles are the way that scientists talk to each other. These are not written for the general public. They're not written for the press or for teachers. They're written for scientists, and not only for scientists, but for scientists within their discipline. Meaning there's going to be jargon, there's going to be specific language that when you first begin reading within a particular discipline, you're going to have to build your own glossary and start building up that language. There's usually a pretty steep learning curve when you first get started with it for that reason. But keep in mind that this is the way scientists report to one another about the experiments that they're doing and what they're finding as they're doing these experiments. These articles go through what's called a peer review process. Sometimes this is referred to as the peer-reviewed literature, meaning that if I do a bunch of experiments in the laboratory, I write it up as a paper, I submit this paper, this manuscript to a journal, say Frontiers in Microbiology, like this one here. It's going to first go to an editor who's going to look it over and make sure that they believe it's appropriate and it looks like it was well done and well written, and then they're going to send it out to multiple, usually 3, anonymous reviewers, people who are in the general discipline who can have some oversight. They can ask tough questions. They're going to really put it under the microscope, pun intended, and try to make sure that it's really high quality work, that there's enough replicates, that that the methods were appropriate, that the conclusions that are being drawn aren't too speculative, right? Scientists can't, through this process, catch fraud. We need really other systems in place to catch that on the incredibly rare occasion that happens, but we can catch mistakes, we can catch sloppy work, and so this whole peer review process is a way of vetting the research before it gets out. There's no 100% guarantee stamp that it's perfect, but this is definitely the highest quality materials, meaning that if you have what it takes to read these articles, you don't have to read a review article or a magazine article or something in what we would call maybe the secondary or the gray literature, where there isn't any oversight, there isn't any accountability or peer review. This is going to be the best quality material, best quality information that's been vetted to the best of our ability. So how is an article like this organized? Well, you're going to have the title of the journal on the front page, and then you're going to have the title of the article. And the title of the article is really carefully selected. We'll talk more about that later. But hopefully the title of the article can give you immediately a sense of whether or not you want to read the paper, and whether it's what you're looking for as you're searching for papers, whatever your purpose happens to be. And then hopefully a really good sense of what this article is about. So in this case, characterization of 4 multi-drug resistance plasmids captured from the sediments of an urban coastal wetland, right? A lot of keywords, a lot of buzzwords in there. You can tell characterization, it's not necessarily hypothesis testing, it's going to be more descriptive research. 4 multi-drug resistance plasmids. So we're talking about antibiotic resistance. We're talking about plasmids, the elements that that can cause or confer drug resistance to bacteria. How do we know it's bacteria? You kind of need to know a little bit about the field, right? For one thing, we're in a microbiology journal. That doesn't necessarily mean bacteria, it just means microscopic organisms. But then in the world of microscopic organisms, 99.999% of all plasmids are bacterial. We have seen a couple weird little plasmids in eukaryotes, but this is really going to be a bacterial phenomenon. And then it says that they're captured, telling you that they're novel, from the sediments of an urban coastal wetland. So These actually came out of nature as opposed to, say, an infectious pathogen. And you got to piece all that together and ask yourself, is this going to tell me what I'm after? Is this part of the story that I'm trying to educate myself on? Or what's my purpose for reading this paper? Do I want to go any further? Then you get into the list of authors, and as you become an expert in a particular area, you're going to find as you read more and more and more articles that you're going to start seeing a lot of the same names. Because although microbiology is huge, and there's tens of thousands of microbiologists around the world, maybe hundreds of thousands, within a particular subdiscipline, let's say multi-drug resistance plasmids, there's going to be a smaller collection of scientists who are actively doing research and putting out papers, and you're going to get familiar with the universities that they work at, like it says down here. Then we go into an abstract. Sometimes it'll actually be labeled abstract. Other times it's just going to jump right into a long paragraph. When I say long, it's usually about 250 words ballpark. There are some journals that go smaller. Very rarely will you see it longer than 250. Some journals will break it down by intro, result, or methods, results, discussion, so a little mini summary of each of those sections. More often you're going to see it as a large paragraph like this that really is an executive summary of the entire study. You really want to get good at reading abstracts because, again, that'll tell you if you want to continue reading any of the rest of the details of the paper. I wouldn't recommend just reading the abstract and making important decisions based on that. Right? You're going to want to, if the abstract tells you this is good, dig in deeper to the rest of the sections. So what are the other sections? Well, there's some kind of an introduction usually that will set the stage for the question they were asking or the hypothesis they were testing, right? It didn't just come out of the blue. What led them up to this? A journal article really does, in many ways, follow the logic of the process of science or the scientific method. There needs to be a reason for a rationale for the question you're asking and the hypothesis that you're testing, and that is set up in the introduction. And then almost always, authors will, in the final paragraph of the introduction, tell you the purpose of this particular study in light of all these other things. So in light of all this stuff that we know, this has led us to this particular study. Okay, so it may say the aim of this study or the purpose of this study, or it may specifically say our hypothesis was, or we asked the question. But that last paragraph is going to give you the goals, essentially, of this particular study, and the rest of the intro was the context of how they came up with what it is they're trying to accomplish here. Then we're going to get into materials and methods. This is really nitty-gritty details. Unless you need to know exactly how a method is carried out, and sometimes that's why you're reading an article, but unless you need to know exactly how it's carried out, often you can just skim the materials and methods so you have a mental map of what types of techniques were used without every last detail of how many microliters of this and how many micrograms of that. Now, if you're trying to learn a new technique or compare their technique to a method you're using that maybe isn't working right, and so you want to modify a little bit, you really got to get into that nitty-gritty in the materials and methods. You can see subsections, right? Description of the study site, antibiotic abbreviations that'll help you to read through the rest of the paper. Endogenous plasmid capture. How did they capture the plasmids? How did they capture them exogenously? You have to figure out what that even means. How did they mobilize the plasmids? To get them to move? How did they do their antibiotic susceptibility testing? How did they determine plasmid copy number? How did they sequence the genomes and then annotate and analyze those sequenced genomes? And then if I want to go look at those sequences myself, what are the accession numbers in the GenBank database that I can go to? All that information that you need theoretically to repeat the experiments yourself should be there in great detail. And then you get into the results. Some journals will mix results and discussion. More often, it's more common practice to have results where you're simply reporting, here's what we got, and then in the conclusions or discussion, you're going to discuss the relevance, the meaning, the significance of it. So here we've got a results section, little intro paragraph, and then again some subheadings that'll help kind of guide you through the story. Remember, there is a story being told here, and there's a logical sequence to this story. And so even if you're not going to read, say, the methods and materials in great detail, you want to actually skim through it so you know the logical progression of the story. So here they describe a plasmid, PLNU11. Now we start to get, and this is where we get into the really good stuff, tables and figures. Now this isn't their raw data, right? They didn't just give you every last measurement that they took. This is summarized raw data that gives you the most important things like means and standard deviations of replicate values, the size and base pair, the capture method, right? Here are maps of plasmids. They're not going to give you the entire 143,600 base pair sequence here. You can go to GenBank to get that raw data. It would be a waste of publication space to just stick a string of A's and T's and C's and G's here for you to analyze. The text within the results, very important to read, and then you're really going to often camp out and spend a lot of time on the figures, reading the legends, looking carefully at the details to learn what you can from them. More figures, more tables. We're telling a story. Remember, how's this story playing out? You knew the steps involved from the methods and materials. Now, how did the results kind of play out? And then most often, you're going to have a discussion section. Sometimes it's called conclusions. Sometimes it's called discussion and conclusions. Sometimes there's actually discussion and a conclusions. Most commonly, it's a discussion section that includes sort of pondering the data, thinking about the relevance and significance of the data, and maybe speculating a little about next steps, new hypotheses, where it might go. And then finally, in the end, you're often going to see things like which authors did what work, where did the funding come from, who helped us out that their contribution didn't rise to the level of authorship, but we want to make sure that they know we really appreciate the work they did. And then we get into the references and supplementary material. More and more we're seeing digitally data, figures, sometimes even the raw data we talked about, pushed online someplace so you can access that. So keep an eye out for supplementary material. And then this reference section is an absolute gold mine for you, especially as you begin working in a new discipline, because you may read something way back here about CTXM class commonplace ESBLs rendering cephalosporins useless, and you're like, I really need to know more about how CTXM class enzymes work, and you've got some references you can go to, Patu and Lopland, and you go here and you find that paper, and you can right here and you can go look it up, and all of a sudden you're on a trail. And rather than it being a single trail, it's more like a web or a network of trails. And you're going to be able to really begin learning. And this is where self-learning comes in, right? No one's giving you a lecture on this paper. It's not college anymore. You've got to be self-taught. And so you're going to have to go dig up that information. And this reference section here sends you maybe only to one other paper, but then that paper sends you to 2 others. And those 2 each send you to 4, and pretty soon you've got this web or this network of papers. So this is how a primary article is written. This is why it's written. This is how it's typically organized. And in subsequent videos, we'll talk more about what's going on in each of those sections. So good luck as you guys read these articles. Maybe for the first time, you're going to really enjoy it.